Welcome back to another video where we will continue our discussion of MHC and now turn our attention to genetics of MHC, the way that it is encoded within the human genome, and how some of the special features of the genetics of MHC sort of solve the diversity problem in terms of all of the different unique antigens that need to be presented. How does the way that we encode MHC sort of get around that problem? Because as we'll see, MHC, even though it needs to bind to so many unique antigens, just like TCRs do, MHC does not use somatic recombination. There are other genetic strategies that we use for MHC to make sure that we can account for all of the possible antigens that need to be presented and to sort of match that really diverse TCR repertoire that we put together in our last set of videos. So jumping into the gene structure of MHC, I want to introduce some terminology that will be helpful as we understand some genetic principles at play, particularly that are particularly important for MHC. The first is the idea of polymorphism. Recall that for basically all genes that you have, that you express in your body, that you have two copies of those genes, one on each chromosome, one on the paternally inherited chromosome, and the other on the maternally inherited chromosome. And so you can see here that what is being represented is that this is a gene that has a unique allele for each chromosome. There's a red allele and a yellow allele. The fact that the cell is heterozygous for this particular gene says that the gene is polymorphic in whatever species the cell is derived from. And so we'll just assume that it's human, and we'll assume that what we're looking at is MHC. So this particular MHC gene exhibits polymorphism because there are distinct alleles that not only it expresses, it is heterozygous, but again, at the population level, it suggests that there are multiple versions, multiple alleles that could be inherited. And so MHC is actually the most polymorphic set of genes in the human genome. MHC exhibits the highest degree of polymorphism out of all genes. And we'll see why that is so particularly important and so particularly adaptive for MHC as we continue this discussion. For now, I want to contrast polymorphism, which again has to do with the existence of multiple unique alleles that might be inherited, with a related but distinct concept called polygeny. Polygeny, meaning multiple copies of the same gene. So here we have these MHC molecules, and this cell is actually showing that it is expressing multiple copies of the MHC gene. So there's a red copy in which itself has two alleles. There's a green copy which has two alleles, and a blue copy. And so this represents three distinct genes, all of which encode the same protein, or at least highly similar proteins. But we call this polygenic because there are multiple gene copies, which in this case all functionally encode their own version of the protein. So it's the same kind of type of protein, but maybe it'll be slightly different between each individual gene. And you can actually see here that for these three copies of the gene, this cell is actually homozygous at each position. And so if we assume that the colors just denote the different genes and not different alleles, we might even call this cell polygenic, but monomorphic, because we don't see different alleles represented for each particular gene. But if we put these two concepts together, polymorphism and polygyny, you can see a cell that is both polygenic and polymorphic, meaning that it has three copies of the gene and that it is heterozygous at all three positions. There are unique alleles represented, and in fact, this represents in total 6 unique alleles encoding this MHC molecule. What's so important is that the expression of 6 unique alleles makes it possible to express 6 unique protein isoforms on the surface of the cell. So assuming that these alleles are genetically distinct enough, they're going to encode unique polypeptide sequences, unique protein sequences that are going to fold slightly differently and give us slightly different protein isoforms, which in the context of MHC means that we're going to have different MHCs that are going to be able to bind to a unique set of antigens. So one thing that's I should point out here is that we saw that with TCRs, they need to bind to a set of unique antigens, but they sort of solve that problem in a different way. Not only differently in terms of their genetics, they use somatic recombination, but also TCRs tend to be incredibly specific to a single antigen, or at least to a very small group of extremely similar antigens. There's more of a one-to-one correspondence between TCR and one unique shape that it happens to be complementary to. MHC is much more promiscuous than that. MHC molecules themselves can hold on to many more distinct antigen types. But remember that there, are astronomical numbers of unique polypeptide sequences. So for TCRs, the theoretical number that we can make is like 10 to the 15th because there are so many unique antigens that we might need to recognize. We don't make anywhere close to 10 to the 15th unique MHC molecules. In fact, you can see that in this example, there are 6 of them on the surface of the cell. So instead of making a sort of one-to-one correspondence of MHC with antigen, MHC instead is more promiscuous, but nevertheless, there's just no way that we would ever really have an MHC molecule that on its own, as a single shape, would be complementary to all of the many, many billions and trillions of potential antigens that it might need to bind to. So instead, even though MHC molecules are promiscuous, no one is going to be promiscuous enough. So we encode multiple copies, multiple distinct isoforms of MHC, all of which hopefully are slightly different from each other, and therefore they themselves are going to present a slightly different set of peptides to T cells. And so if we get enough unique isoforms, and they're all distinct enough from each other, that gives us sort of good coverage of each of them having their own sort of unique turf that they cover in terms of the peptides they can bind to. And hopefully, and as it turns out in practice, this is true, almost any peptide that we need to present, that we might encounter and process in our bodies, at least one of our MHC isoforms is likely to be compatible with and being able to present. And so the degree of polymorphism for MHC genes makes it so that we tend to accumulate many unique alleles, which will give us many options for different isoforms. And then the fact that we encode MHC polygenically, again, gives us the opportunity to express at the protein level many more distinct isoforms than we would under normal conditions. So the combination of polymorphism and polygyny are very important ways that the genetics of MHC sort of, again, address the diversity problem in terms of all of the potential antigens that might need to be presented. So having presented or explained these two important concepts, polymorphism and polygyny, let's actually look at how they apply to MHC in particular by zooming in on a region of the DNA in humans that we call the human leukocyte antigen complex, or HLA complex. The HLA complex is a large region of chromosome 6 in humans, which we can subdivide further into a class 1 region, class 2 region, and a class 3 region. And these will roughly correspond to MHC class 1 and MHC class 2, as I'll get to in just a second. But first, I would like to address this sort of unexpected member here, the class 3 region. So to this point in the course, I've only ever referred to MHC class 1 or MHC class 2. And I want to explain here that there is something called the class 3 region within the HLA complex, but it encodes important immune proteins that themselves are not really MHC molecules. They tend to be involved with innate immunity, molecules associated with the complement system or TNF family cytokines, but molecules that do not interact directly with peptide or present peptide to T cells. So because of that, they are not really the focus of our lecture today, but I at least want to make sure you know that there is a class 3 region, but there's no such thing as class 3 MHC that presents peptide. So we'll focus on the ones that are more germane to the points that we want to make today, which are MHC class 1 and MHC class 2, both of which are encoded within the HLA complex within regions that bear their name, the class 1 region and the class 2 region. Let's focus on the class 1 region first. I said that MHC genes are polygenic, and here we can see that play out. Within the class 1 region, we actually see that for MHC class 1, we encode 3 distinct genetic isotypes of MHC class 1, meaning that there are 3 genes for MHC class 1, which are designated A, B, and C. The full gene names then are HLA-A, HLA-B, and HLA-C. These single genes encode the alpha chain of MHC class 1. So recall that MHC class 1 is made up of a single alpha chain, and that alpha chain is the subunits alpha 1 and alpha 2 are what make up the peptide binding groove, which you can see here. But that alpha chain needs to complex with a molecule called beta 2 microglobulin, which is being represented by this kind of gray square here. Beta 2 microglobulin is actually not encoded in the HLA complex. We are actually right now only talking about the encoding of the alpha chain, which is the more important part that interacts directly with antigen. And so there are three genes that will encode three isotypes of that alpha chain, and they get these names A, B, and C. So already you can see that this is going to give us three isoforms potentially of MHC class 1 proteins that might be expressed on the surface of a given human cell. For class 2, things are slightly more complicated, but the same general trends apply. There are three isotypes of MHC class 2, which we call DP, DQ, and DR. But unlike MHC class 1, MHC class 2 is a dimer of alpha and beta chains, both of which interact with antigen, both of which are required to form the peptide binding groove. So within the class 2 region, we actually have 2 genes per isotype, and each of those genes encodes an alpha chain and a beta chain. So you can see here that to make a functional HLA-DP protein, you need a gene that's going to produce a DP alpha chain, and then another gene that will produce a DP beta chain, and those two things are going to dimerize after they have been translated. The dimerization, though, I should point out, does not occur promiscuously across isotype. What I mean by that is that DP alpha chains will only dimerize with DP beta chains, and you don't see cross dimerization, for example, of a DP alpha chain with a DQ beta chain. The chains always dimerize within their isotype, DQ to DQ, DR to DR, and so on. The end result of that is also that we end up with, where at least we're starting out now because of polygeny, with 3 potential protein isoforms of MHC class 2. Again, hopefully all of which will bind a different set of potential antigens, although right now we haven't quite gotten to why that's possible yet. But here you can see that the gene structure is polygenic, at least for MHC class 1 and for MHC class 2, those peptide binding versions of those molecules. So if we look in more detail at the HLA complex, we can see some additional features emerge which are worth discussing. So here, remember, we're in human chromosome 6, and we're now looking at the entire HLA complex and the way that it actually unfolds, or the way that it is actually encoded in terms of the order of genes and where their positions are on the chromosome. So you can see actually that if we go from centromere to telomere, that it is the class 2 region that is encoded first, followed by the class 3 region, and then finally the class 1 region. Let's start by looking at the class 2 regions. Within the class 2 region, you can see highlighted in blue what we would call the classical class 2 MHC genes. Again, those isotypes DP, DQ, and DR. They are classical, or called classical, because they are the peptide presenting MHC class 2 genes. They're the important ones that we've discussed to this point in the course. But notice there are some other genes shown here in black. We see DO and DM. They have similar sounding names to DP, DQ, and DR, but they do not bind to antigen directly. They have other regulatory functions, which we saw in a previous video, had to do with peptide loading, but not peptide presentation. And so for that reason, we think of them as non-classical MHC genes, and that's one reason why they're here shown in black, because they sort of have a different type of function. In addition to those molecules, there are also some accessory proteins that are involved with endogenous antigen presentation as well. Molecules or genes such as these LMP genes, which are actually proteasome subunits that are important for making the immunoproteasome, which was important for degrading ubiquitinated proteins in the cytoplasm. As well as we also see within the class 2 region molecules like TAP, which is important for transporting endogenous peptides out of the cytoplasm and into the ER. This is a little bit, potentially confusing, because these molecules are so important for endogenous antigen processing, and these are important for the process of putting antigens onto MHC class 1. But notice that they are within the class 2 region. This is just a little bit of a discrepancy that you You just sort of have to know. But it means that loss of the class 2 region, for example, if you had some kind of genetic deletion of the class 2 region, would not only impact exogenous antigen presentation, because you'd be lacking things like the class 2 isotypes as well as DM and DO, but it would also impair endogenous antigen presentation, because you would be lacking these important molecules associated with the immunoproteasome as well as the peptide loading complex. So moving down the line from the class 2 region, we can see that within the class 3 region, we have some innate immune molecules, as I said, like C-reactive proteins, as well as TNF family cytokines. But moving right along to the class 1 region, which again is more important for our lecture today, notice that there are also 3 genes that are highlighted here in red. Those are HLA-A, B, and C, which again are the classical class 1 MHC genes that actually present antigen, as well as some non-classical genes, HLAs E, F, and G, which again we haven't talked about to this point, but also just have immunoregulatory functions that do not involve the direct presentation of antigen. But it's worth knowing that they are within the class 1 region and that they have their own roles to play within the immune response. It's just different than the classical genes A, B, and C. So these are the genes, and again, this is to underscore that the antigen-presenting MHC molecules are encoded polygenically. We have 3 isotypes of each, and for MHC class 2, we actually have genes for each chain, as I said before. But one thing to keep in mind is that here we are looking at a single chromosome. But remember that humans have 2 chromosomes, or 2 versions of each chromosome, and that there's another chromosome 6 that is not represented here. So if we imagine that what we're looking at right now is the paternally inherited chromosome, unpictured is the identical maternally inherited chromosome 6, which means that for all of these genes, there are actually 2 copies and potentially 2 distinct alleles for all of these genes. And those alleles may not be identical between the maternal and paternal gene copies. And if they are not identical, then we would say that this particular person was heterozygous for whatever those particular genes might be. And so the degree to which any of the genes that you see here on this slide are polymorphic changes depending on whether or not we're talking about a classical MHC gene or a non-classical, those that present antigen versus those that don't. And so if we want to sort of summarize that conceptually, you could get an image that looks something like this. If we start within the MHC1 region and we look at the classical versus non-classical MHC1 molecules, let's start with A, B, and C, which are the classical MHC1 genes that actually present antigen. Notice here that they are depicted in red, indicating that they are highly polymorphic. And in fact, the class 1 alpha chain genes are the most polymorphic genes within the entire human genome. And that's important because they have a lot of different peptides that they might need to bind to and might present to CD8 T cells. And so the high degree of polymorphism, again, is important because it gives us more options, more diversity in terms of the antigen binding repertoire that we need to sort of encounter or account for. But notice that while the alpha chain is highly polymorphic, this yellow square here is meant to represent beta-2 microglobulin, which is more structural, and it is actually monomorphic. There is no evolutionary advantage to having many different forms of beta-2 microglobulin because it's just a structural molecule. And in fact, it's probably better only to have one version because it works and you can be sure that it works. And because it's not interacting with antigen, there's no need for it to be different from one version of MHC class 1 to the other. So beta-2 microglobulin is monomorphic in comparison. The other comparison we can make are the non-classical MHCs, E, F, and G, and as you can see that they are not highly polymorphic, but they are either oligomorphic, meaning there's sort of a handful of potential options, or monomorphic, meaning there's really only one common or functional allele, or at least isoform that tends to be expressed. And again, we haven't really talked about the function of these molecules, but it is not, they do not involve direct interaction with antigen. Therefore, there does not need to be such a high degree of variability for these non-classical MHC genes, and therefore evolution has not favored high degrees of polymorphism for them. If we look at MHC class 2, we see similar principles unfold. If we look at the non-classical MHC2 genes, DM and DO, which remember were regulatory in nature, they involve the swapping out of CLIP for peptide in the process of antigen loading, but they themselves do not present antigen directly. They also have low degrees of polymorphism. You can see here their chains are either blue or yellow, representing oligomorphic or monomorphic frequencies of alleles within the population. However, if we look at the classical versions, DP, DQ, and DR, these three isotypes of MHC class 2, we see that in comparison, they are much more polymorphic. DP and DQ are both chains are highly polymorphic. DR is a little bit of an exception to the rule. The alpha chain is not particularly polymorphic, but the beta chain is extremely so. And so the beta chain, the fact that the beta chain is so polymorphic sort of makes up for the fact that the alpha chain is a bit less so. But nevertheless, the general trend that the MHC class 2 molecules that bind to antigen, DP and DQ and DR, are much more polymorphic than their non-classical counterparts. So this conceptually makes the point of which ones are polymorphic and which ones are not and why. But let's put some numbers to these concepts just to sort of underscore the point more fully. So these are numbers that I actually pulled from publicly available data. They are fresh off the presses, at least at the time of recording of this video in October of 2025. So these are actually currently all of the known alleles for classical versus non-classical MHC class 1 genes. I could show you a similar table for MHC class 2, but because there's so many more genes there, this is just a little tidier. So we'll just focus on MHC class 1 to make this point. But notice that for the classical MHC class 1 genes, A, B, and C, that there are an incredible number of alleles represented in the human population. 9 or 10,000 basically, again, right now, and new alleles are being discovered all the time. This is a very high degree of polymorphism. As I said, this represents the most polymorphism out of any genes that we find in the human genome. But if we compare it to the number of known alleles for the non-classical MHC1 genes, E, F, and G, notice these numbers are much smaller, orders of magnitude smaller, essentially. Again, that's because there is no reason to have so much variation in these non-classical genes because they do not need to interact with all of the possible variations of antigen that the classical MHC genes need to. Another point that I want to make here is that there's a distinction between the number of alleles that exist, meaning the set of unique genetic sequences that might constitute a given allele, versus the number of unique protein allotypes that are functionally expressed. An allo meaning different, an allotype is an actual distinct protein conformation. And the reason that this number is somewhat smaller, still very big, but is somewhat smaller, is that many alleles are actually synonymous with each other. Remember that an allele is just a DNA sequence, but for DNA to become protein, we sort of read it as a set of codons, and that many codons are synonymous. They encode the same amino acid. So the degree of codon this is, the synonymous nature of many codons, the result of that means that many alleles end up producing a functionally equivalent protein. And so you can see that the number of unique allotypes then of non-synonymous alleles essentially is smaller but still very high, still thousands of unique allotypes of MHC class 1. So this still gives us lots of options, many unique potential combinations of alleles within an individual person, which hopefully again will give them a unique combination of isoforms on the surface of their cells. And so what does it matter that there are so many different allotypes? Or I should say, alleles represent sequence diversity in terms of the DNA that encodes each of these isotypes of MHC. But where does that sequence diversity occur? And is there a logic behind where within the protein tends to be most different from allotype to allotype? And how does that influence antigen binding? Well, if you actually take MHC class 1, again, we'll focus on MHC class 1 for simplicity's sake here. If we take the molecule of MHC class 1 and we unfold it and we stretch it out and we look at its primary amino acid sequence from the beginning to the end, you can see here that MHC class 1, is about, I guess, 280 amino acids long. What you can see here is that if you look on the y-axis, which is just graphing the amount of variability that you would expect, or that has been recorded when we compare allele to allele, we see that certain amino acids tend to be more different than others when we compare any given allele to any other given allele. And that the frequency of variability is kind of enriched at certain amino acid positions. So for example, kind of amino acids 41, 2, and 3, I guess, here tend to be more different than the amino acids between 20 and 40. And if you look at the amino acids that show the most variation among alleles in the human population, you see that those amino acids, where they actually end up in the folded tertiary structure of the MHC class 1 molecule, is actually right in the peptide binding cleft of the molecule. So they actually sort of form the lining, they form the kind of shape of that basket structure that the peptide actually sits on and interacts with physically. And that makes perfect sense. That is the portion of MHC that we need to be different, that we need to be diverse from allele to allele. And so where alleles tend to differ from one another are at those positions that encode the amino acids that make up the peptide binding cleft. Whereas the amino acids, again, that are more structural in nature, especially those associated with the alpha 3 subunit, they show a lot less variability from allele to allele, because the structural portions, they actually need to relatively be more conserved, because the structure, there tends to be a sort of single way that structure is most functional, and the variability then is left to the portions that are going to interact directly with antigen. So there's a final point that I want to make about the gene structure of MHC, or at least a feature of MHC genetics, which sort of allow us to harness the polygenic and polymorphic nature of MHC expression to make the maximum number of unique protein isotypes on the surface of our cell, and that is the idea of codominant expression. So classical MHC genes, very importantly, are codominantly expressed, meaning that every allele at every gene will be functionally translated into a unique protein isoform that will be expressed on the surface of the cell. This is different than many genes, and especially some of the genes that you may have learned about in previous coursework in genetics. Many genes very famously have alleles that are dominant and those that are recessive. And in that case, it is only the dominant allele that is expressed, while the recessive is not expressed. Or there are some cases where particular genes are allelically excluded or silenced in some way, and where one allele will always dominate. That is not the case for MHC genes. They exhibit codominant expression, meaning that whatever two alleles you have for any given gene, both will be functionally translated into a protein. And as you can see here for MHC class 1, remember that there are three gene isotypes, A, B, and C, and that there's a paternal and a maternal allele for all three of those genes, giving you 6 total MHC class 1 alleles. And assuming heterozygosity for all three genes, meaning that this individual has 6 unique alleles, which in fact is what most people have. Most people have 6 unique alleles. And remember that there are like 10,000 options, so it's really not that surprising that most of us are heterozygous at all three positions and have 6 distinct alleles available to be expressed at the protein level. And so most of us then, following that logic, can actually produce 6 unique isoforms of MHC class 1 on the surface of our cell. And these isoforms, because they all come from distinct alleles, are all going to have a slightly different shape, and the peptide binding cleft of each of these isoforms is going to have a slightly different shape, meaning that their ability to bind to certain antigens is going to differ from one to the next. And hopefully there's enough diversity among this 6 that whatever endogenous peptides that we might need to present to a given CD8 T cell, that at least one of these isoforms will be compatible with any peptide that we might encounter and need to present. And in fact, that is generally true for most of us. And that is the power of the combination of high degrees of polymorphism, along with polygeny, and the exhibition of codominant expression that gives us a nice and differential set of protein isoforms, which will give us good coverage against all the possible antigens that we might need to present. And the same thing essentially is true for MHC class 2, although it's slightly more complicated, as you can see here. The main difference is that MHC class 2 requires dimerization of alpha and beta chains, as we've said. And those alpha and beta chains are each encoded by their own gene. And across the three isoforms, DP, DQ, and DR, that means that there are 6 classical MHC class 2 genes. And we express, again, 2 alleles for each. So there's up to 12 unique alleles associated with MHC class 2. And it just so happens that these 12 alleles also result in 12 unique isoforms of MHC class 2. Although it's not that one allele equals one isoform. There's a slightly more complicated thing going on here, which I would like to unpack. Remember that MHC class 2 requires dimerization of an alpha and a beta chain. So if we focus on the DP isotype here, what I want to point out to you is that alpha and beta chains can be formed using the chains that come from either the paternal and or the maternal chromosome. And so to explain that a different way, you can see that you can make DP MHC class 2 protein using the beta chain that was encoded on the paternal chromosome, dimerized with the alpha chain that was encoded on the paternal chromosome. Or you can use the beta and alpha chains that were both encoded on the maternal chromosome, or you can mix and match. You can use a beta paternal with an alpha maternal and vice versa, beta maternal with alpha paternal. And these are what constitute the actual isoforms of MHC class 2. And so that matrix of paternal to maternal sort of dimerization will give you 4 functional possibilities. And that assumes that you are heterozygous for all 4 alleles. As long as you are heterozygous for all 4 alleles, you will make 4 unique isoforms of MHC class 2 within each isotype. And so again, assuming heterozygosity across the board, you add 4 to 4 to 4, and that means that the average person, who again is going to be a heterozygous at all of these positions, will have 12 unique isoforms of MHC class 2 on the surface of their cells, giving them, again, broad coverage now to present a very broad set of exogenous antigens to CD4 T cells. Okay, we can now summarize our discussion of the gene structure of MHC. We saw that MHC diversity is also very important, just like TCR diversity, for sort of giving us coverage against all the broad possible antigens that we might encounter. But instead of relying on something like somatic recombination, MHC is instead, it addresses the diversity problem through polygenic encoding, as well as a high degree of polymorphism in the population, which gives us many potential variant isoforms that we can express at one time. MHC, classical MHC genes, as well as other accessory molecules, are encoded in a large region, a large contiguous region of DNA that we call in humans the HLA complex. And within the HLA complex, the most important genes that we covered today were 3 isotypes of MHC class 1, which were given the names HLA-A, B, and C, as well as 6 unique genes, 6 unique loci within the MHC class 2 region, each of which encode the alpha and beta chain of 3 isotypes, DP, DQ, and DR. Finally, the combination of all these concepts that we've gone over here gave us a lot of potential diversity in our antigen presentation repertoire. All of the allelic variation mattered because it influenced the shape of the antigen binding grooves of all of the potential isoforms of MHC. So they're going to all bind to a different set of potential antigens. And the fact that MHC molecules are codominantly expressed sort of doubles the functional number of isoforms that we can express at one time, which again gives us more coverage against all the potential antigens that we might want to present in the context of any particular infection. So this covers it for our discussion of the gene structure of MHC. And our next and final video in this module, we will look at a more sort of conceptual level at some other considerations of the genetics of MHC, particularly the evolutionary genetics of MHC in humans. I'll see you in that video.